So because we've been sponsored for the by the National Science Foundation, they also are giving us the support to reach out and present this group. I'm going to, let's see, next, we really owe a lot of thanks to Brian McMullen, who he and his family run this pub, and their wait staff. They're absolutely phenomenal. Wait staff, Cassie and Susan, they will field anything. They know the answers to our trivia quizzes if you need somebody to whisper in your ear. And so please go onto Facebook and thank Cassie, thank Susan, thank Brian McMullen for letting us have the Science Cafe here. As Sheila already told you, one of our really great UNLV graduate students, we will not gather in January. It's too close to New Year's. And our next gathering will be the first Wednesday in November. We're going to have a talk on hibernation. Oh, sorry. Thank you very much. I'm totally unable to follow time. In the first Wednesday of February, we will have a talk on hibernation, life below zero, by Frank van Breukelen, a professor at UNLV. So before I introduce our speaker, I'm just going to tell you the order of events. So we're going to have our presentation, then we're going to take a 10-minute bio break. So you can, if you can, squeeze out from your table. We'll have 10 minutes for coming and going, so our speaker can also come and go for a few minutes, rest his voice, and then we'll, at the end of that 10 minutes, we'll announce the trivia contest winners. We have 3 prizes. We've placed note cards and pencils on your tables so you can jot down questions as you think of them. At the end of 10 minutes, our speaker and I will come back. You can either give me your questions, put your name on it, and I'll read your question for you, or you can put your name on it and we'll call your name and you can ask your question yourself. So it's your choice depending on how shy you are. I get the feeling we don't have many shy people here tonight. But I'm glad to hear it. So with that, I want to introduce our speaker tonight. It's Professor Martin Schiller. He's a professor in the School of Life Sciences at UNLV. He's also the executive director of the Nevada Institute of Personalized Medicine. And that's what he's going to talk about today, personal genome sequencing, human genetic variation in health. Hold it closer?
Yeah.
Okay. I'll do the best I can. So we asked him how he got interested in doing this kind of research. He actually didn't train in human genetics, and this is often common for scientists. We all start out training in something else, and we follow this circuitous path to whatever we're doing now. And so basically what Marty Schiller did was he hung out with other human geneticists while he was at another university. He got really interested in the topic of human genetics and human DNA variation. And so that is now his topic of research here at UNLV. And that's what he's going to talk to you about today. I think at this point, then I will introduce Martin Schiller, who will give his presentation.
Thank you. Thank you. Thank you for the invite. This is one of my favorite things to do, is to have a beer and speak to a lay audience. Okay. All right, so we're going to start with a question. Sometimes, you know, when you're starting out as a speaker, you ask the crowd a question, you get nothing but silence. So let's see if this works. So would anyone in the room pay $100 for 15 extra years of life expectancy?
I might pay $100.
How many people? We have a naysayer.
$100 a year.
Guarantee that. Rough metric. Might be good.
Is my mother-in-law still
take a
around?
chance. No, you can live without your money.
$5.
All right, well, toast the person next to you because, raise your glass and toast the person next to you because you've already bought it. Believe it or not, you've already paid for it and you're getting it today. So I collected some data from the web about 10 years ago and put together this plot. It's the life expectancy over time over the last 4 millennia. It's the blue curve, and if you notice, in about 1800, the life expectancy of a human starts taking off drastically, just at about the time of the Industrial Revolution, and also fueled by scientific research in the area of biomedicine. So if you calculate out, you'll have to trust me, if you fit the last part of that curve to a line, for every year you live, you get 2.2 additional months of life expectancy. And how much has this costed the U.S.? So the NIH was formed in the 1940s, and they started heavily funding the scientific research. And the total cost is about $1 trillion we've spent to date. So less than 1 year of the U.S. budget. If you calculate out your 70 to 80 year life expectancy for an extra 15 years, and that number, it's costing you about $100 a year. So you've already gotten that. Isn't that cool? Right? Is that the best buy in history? What? It is. We have a naysayer. Why not? What do you want to buy for the $100?
No, it's a causation correlation fallacy. And actually, if you look at water systems, you plot against water systems, it works much better.
Okay. So maybe it's not entirely due to biomedical research, but from a personal experience, there's a lot going on where medicine is increasing life expectancy. There could be other contributors. All right, so if we want to kind of figure out how we might do better than that or continue the curve, it's probably good to look back at the history of, do you want to turn that on?
Yeah.
It's good to look at the history of where medicine was. So if we look back maybe 1,000 years at what medicine was like, I found this video that accurately depicts what medicine was like about 1,000 years ago. So in 1303 AD, well, I guess 700 years ago.
Do you want the beginning too? Like,
No, this is fine, just get the volume high.
Hello, Theodoric of York. It's springtime, and I'm here for my annual haircut and bloodletting.
Well, welcome, son of Miller, William. Oh, it's time to see you now. Have a seat. Broomgilda. You start on his hair, and I'll open up one of his veins.
yes, Theodoric!
It's an
How's
intervention.
that little baby I delivered last Christmas when your wife died?
The little fellow is deformed.
That's right. I remember now. This may hurt.
And now it's time for another episode of Theodoric of York, Medieval Barber.
There you go. Well, looks like I have another patient. I'll be back in a minute to see how you're doing.
Right, thank you. Just a trim, Broomgilda.
Hello, Theodoric Barber of York.
Hello, Joan, wife of Simpkin the Miller. Well, how's my little patient doing?
Not so well, I fear. We followed all your instructions. I mixed powder of staghorn, gum of Arabic with oiled sheep's urine and applied it in a poultice to her face.
And did you bury her up to her neck in the marsh and leave her overnight?
Oh, yes. But she still feels as listless as ever, if not more.
Well, let's give her another bloodletting. Broomgilda,
Yes, Theodoric!
take 2 pints.
Yes, Theodoric!
Will she be all right, Barber?
Well, I'll do everything humanly possible. But unfortunately, we barbers are not gods. You know, medicine is not an exact science, but we're learning all the time. Why just 50 years ago, we would have thought your daughter's illness was brought on by demonic possession or witchcraft. But nowadays, we know that Isabel is suffering from an imbalance of bodily humors, perhaps caused by a toad or a small dwarf living in her stomach.
That's a diagnosis.
I'm glad to see she's in such good hands.
Thank you.
Oh, God. Theodoric, borrower of York?
Say, don't I know you?
Yeah, you worked on my back.
What seems to be the matter with your friend here?
I broke his legs. I was at the festival of the Vernal Equinox, and I guess I had a little bit too much meat, and I darted out in front of an ox cart. And all happened so fast, the poor little fellas couldn't stop in time.
Well, you'll feel a lot better after a good bleeding. Say, who's the barber here?
Okay, just do something for my legs, could you please?
Okay, well, look. No! We'll get him up on the gibbet over here. This might hurt a little bit.
My thing is hurting now.
A little pain never hurt anyone. Okay, he's still up there. Look, what we're doing is separating the bones a little bit. If you don't feel better, we'll just cut them off here.
I'm pretty sure I'll be feeling better tomorrow.
We still do amputation today.
This will teach you to go easy on the knee. Brunhilde put a few leeches on his forehead. Okay.
Thank you, Brunhilde, you're so kind.
How are we doing?
I feel faint, but
Well, when was the last time you came in for a worming?
I guess I'm due, but I don't have time today. Please, accept my payment. This fine fat goose.
Oh, well, thank you. Brunhilde will give you your change.
Thank you, Theodore.
You don't get change today.
Well, how's the little patient doing?
She's worse. She's looking pale.
Well, if she's not responding to treatment, we'll have to run some more tests. Brunhilde, bring the caladrius bird.
One of the first diagnostic
Yes,
tests.
the caladrius bird is a test. We put it next to the patient, and if it looks at the patient's face, then she will die. If she looks at her feet, then the patient will live. So, a little test. Hey! Can you interpret these signs? Seems to be looking up and down there. Well, okay, just take off. Well, I'm not sure how to read that. Just take 2 pints from her.
Yes,
Take 2 pints from that bird, too.
She's dead. Dead! I can't believe my little girl is dead!
Oh, now, Mrs. Miller, you're distraught. You're tired. You may be suffering from nervous exhaustion. Maybe we'll just take some of your blood, too. We'll just...
You're a charlatan. You killed my children just like you killed the rest of my family. Why don't you admit it? You don't know what you're doing!
Wait a minute. Perhaps she's right. Perhaps I've been wrong to blindly follow the medical traditions and superstitions of the past centuries. Maybe we barbers should test those assumptions analytically through experimentation and a scientific method. Perhaps this scientific method could be extended to other fields of learning. The natural sciences, art, architecture, navigation. Perhaps I could lead the way to a new age. An age of rebirth. A renaissance. Nah.
We'll be next week for another episode of Theodoric of York, the Evil Barber. It will be Theodoric saying,
Well, I think a little more bloodletting and some boar's vomit, he'll be just fine.
So it seems rather barbaric and silly what was happening.
I'd like to talk seriously just for a moment.
We don't. Oh, that's it. Oops. But we still do amputation today, and it was invented back then. But think about what medical care might look like if we fast forward 700 years, similarly looking into the past. And I think we're like on the verge of a transformation right now in the approach to medicine, and that's what I think personalized medicine is about. So we need to give a little bit of background first so that we can understand how this is done and what its promise is. So shown here is what's called a karyotype, and it's looking at a cell that was in the process of dividing, and we're looking at the chromosomes, the 23 chromosomes, or pairs of chromosome of a human cell. And each of those chromosomes, if you added them all up, there's a DNA sequence behind them, and that DNA sequence of all 23 chromosomes is about 3.1 billion base pairs long, or units long. So, and that's the code for organisms, and in this case for a human body. So if you look at that, I've kind of drawn out, if you just look at a region on that chromosome, that's what the DNA sequence looks like. It's just a combination of 4 letters which represent chemicals, G, A, T, and C. And that's what we have to work with in the cell. That's the blueprint for life. Now, let's suppose that was the sequence of one person. Well, it turns out that each person has 2 copies of each chromatid, which is called the chromosome. And you get 1 copy from one parent and the other copy from another parent. And if we start sequencing different people, we'll notice a position that's called a variant, or it's an allele, and it's different than the reference. So we have a reference human genome now, which is supposedly representative, and we compare everything to this reference. So there are a lot of these variants in people. You know, like the difference between you and me, there's about 4 million differences in the human genome. So you have 4 million differences between you and the person sitting next to you. And so I also want to kind of think historically about how this came about. So this is a map, and each row is 30 generations. There's been about 6,000 generations since Adam and Eve. The first 4,500 generations were in Africa, and the last 1,500 have been outside of Africa. And include Africa, and we've only been naming people with last names for about 50 generations. And if you look on the right side, there's a couple of key events. The blue arrow would represent when we left Africa. The yellow arrow represents when we started breeding with other hominids. So humans have bred with 2 other species, and actually the only true humans are 2 tribes in Africa. Everyone else is a hybrid with another hominid. So all of us in the room are hybrids with, I would say everyone in the room, with Neanderthals. And then those of us from Southeast Asia and the Pacific Islands are tribrids, because they're part Denisovan as well. And so these introduced new genetic variation into the human population. And so, like, if we look at the orange and how that's expanded, this would be called today a very common variant, because it was 1 base pair position change, like that red allele, a 1 base pair change, and that happened, you know, 78 BC. And, well, 88 BC. And so now it's permeated, and it's present in a whole lot of people in the population today. But there are other variants that were more frequent that were born recently, maybe even in the past few generations, and these are called rare variants. And they each contribute to human health in different ways. And one thing you should know is, if we go back historically, each new person has about 50 new variants. So you have 50 that are pretty much unique to you and maybe a few other people on the planet, but the rest of them are common and rare variants. So the thing is all of these variants contribute to how we present differently with different diseases, and how we respond differently to different treatments. And so you can't just treat everyone the same, which is largely what we've been doing, that things need to be personalized based on one's genetic makeup. And that's the premise of personalized medicine. Okay, so how do we now use this genetics in a way where we can start affecting medicine? One of the common approaches is called a genome-wide association study. And we have several scientists in the audience, Sam and Jing Chan, who are involved in these sorts of studies on humans. And what the idea is, if you look on the left, is let's suppose we can find one of those variants and it basically is not present except at a very low frequency, like shown on the top left. In normal people, that would be gray. But if you looked in a diseased population with any disease you choose, you would find that this variant's highly prevalent. And based on some statistics associated with this, we can determine whether this variant is highly associated with the disease or if it's just by random chance. When we do this, keep in mind that there's about 4 million variants per person, so we're doing 4 million tests. So there is a chance by random chance that we're going to identify a variant that looks like that. But if we do it in a very large set of patients, say 10 to 100,000, then the statistics ends up supporting that that variant really is associated with that disease. And an example is shown in the plot on the right, which is called a Manhattan plot. And as you can see, there are 3 regions of the genome. Each color represents a different chromosome, and each dot represents a different variant. And we do get some variants that have very high statistical probability that they're associated with the disease. And this is for Alzheimer's disease, and the one on right is the ApoE alleles. And they really segregate well in Alzheimer's disease, and they increase the risk of one getting Alzheimer's disease. Now, I'm showing you this is one, but in the past 4 or 5 years, the numbers of this type of study have exploded, and we are all of a sudden today able to associate all kinds of variants with diseases, treatments, and other things I'll go into later. And so this has opened up a really new opportunity in the world for personalized medicine. Okay, so how does that translate into anything like meaningful for us, like doing a treatment? Well, what's shown here is I've taken the same diagram and just added in an arrow because some of the DNA sequences in the chromosomes end up producing proteins, and proteins are going to do all of the molecular work in the cells. And so now, if we had a variant, it ends up producing a protein that's variant, and now if that protein had a function, maybe it doesn't do it as well. So you're all looking at the screen and you have a protein that makes your lens clear called beta-crystalline. And what if you had a variant that maybe made it a little bit less clear? That would be a problem that would present in you. And so that's how you end up moving all the way from just a change in the DNA sequence to a change in one of your traits or in your susceptibility to disease. So now if we look at proteins, it turns out that there's, probably, several 100,000 different types of proteins in the cell. Every gene produces more than one protein, pretty much in general. And if we look at a map of how all of those proteins here represented as yellow dots are connected, inside just one cell in your body, keeping in mind that your body has 60 trillion cells, one cell has this giant network functioning within it. And when you start now adding up the 4 million variants, this network is a little bit different in every person. And so the cell is probably going to respond a little bit different in every person. And this is, again, part of the justification of probably why we can't look at everybody and treat everybody with the same drugs for the same thing or the same surgeries. Now, if we take this out even a step further, I really like this map I created quite a while back, but it's my idea of a kind of a grid of how we even take that information forward. Because if we start on the right side and we're talking about proteins, we're talking about molecules, okay? And all those molecules come together, and I'm just going to shift to the left one cell on the top, and those molecules make up a cell. And all the cells come together and make up a tissue, and they come together and make up an organ, and they come together and make up an organ system, which makes up organisms, which makes up ecosystems. Okay, so there's a hierarchy here, but we're going from a change in the molecules at a very fine level, and it's going to affect an organ system and how it functions, or an organism. I mean, if we're going to have a variant that is lethal, right, we're going really far across this grid and have a change in your genetics that could end up being lethal, okay? So it's a long way away. And what's amazing, and why I hope you're going to hug the scientist next to you when you leave the room today, is that over the past 100 or so years, scientists have published about 25 million articles from research studies that fill in different portions of this grid, okay? And the grid, if we now go down in the Y direction, I think in order to understand the system, you have to understand 6 major things. One of them would be what the system is made of, okay? And I'm going to get away from cells and organisms here and use something simple like a beer tap, okay? So what's a beer tap made of? You know, you basically have a valve, a lever, a tubing, etc. They have a structure. The molecules have structures too. There are physical interactions. The tap has to be connected to the valve when you pull it. There are physical interactions in, like, the protein interaction network I showed you earlier. There are functions. When you pull the tap, it opens the valve. That's the function. Okay, we have valves inside us too. Okay, there are functions, and they're all situated in a particular location. Same in a human, and all of these things change over time. Okay, and that basically, I think, can describe any system, whether it's a human being or a beer valve tap. Okay. What all this does, and this is where the majority of that $1 trillion went, okay, into this grid, is now all of a sudden we can connect a single variant through those GWAS studies to changes in physiology and changes in this grid. And the information is increasing very rapidly. And so all of this work that has been done, we now, through human genetics, have the ability to take advantage of it in the form of medical care. So let's, I think that's it for background. Okay. Are there any questions before I move on? Yeah.
How can you ensure that genetic variation is the cause of a disease like Alzheimer's, not actually the effect of disease etiology?
So that they don't affect?
No. How can you ensure that a genetic variation is actually the cause of a disease like Alzheimer's rather than the result of the disease etiology itself?
Okay. So it's possible it could be an intermediate in a way, but this is where, like, animal models are very helpful. So if you were to now, like, make a mouse that had that mutation and it produced an Alzheimer's phenotype in the mice, right, and the mice had the same symptoms as a human, that would give you a lot more comfort, right, that it's involved. Okay. And so, like, we use model systems to try to determine whether we've identified really the right variant or not. And you don't have to do it in a mouse too. They use lower, more primitive organisms too.
I guess what I'm getting at is, how do you factor in the possibility that the disease itself could cause the genetic variation?
It is a bit of a problem. So like in cancer, the disease itself does cause variation. The genetic material is generally, except in a few instances, extremely stable, right? And so it doesn't change in a disease. But there are cases like cancer where it's a cancer is a disease of the genome where you lose control of the genome. In fact, that the karyotype I showed earlier is actually used clinically because you can see pieces of chromosomes that exchange and move around, and that can be diagnostic for like trisomy and Down syndrome, or for even cancer syndromes. And so neonatal testing involves karyotyping. So there are changes, but it's not in, you know, the majority of human disease here. If you look here, there's 12,000 named diseases. The vast majority of those don't involve changes to the genetic material. Okay, so what are we practicing today? Actually, we were doing it 700 years ago, and if we think back to Steve Martin and what he was doing there, that was trial and error medicine. It wasn't very good, right? Because, well, we never did a clinical trial back then, at least not one that I know of, but, you know, sticking a leech on someone's head and hanging them upside down really isn't likely to improve the health or the condition of someone. But nevertheless, the basic pillars of medicine, such as diagnosis, prognosis, and treatment, were born and were still evident at that time. So if we look, the Caladrius bird, right, that is a diagnostic test. Now, it wasn't a very good one, right? But it still was a test, and we don't use it anymore today. All right? Basically, based on the outcome, he could predict whether someone would die or live. And then he had lots of interventions: blood, worming, amputation, leeches. So we still use amputation today, but what do we use? Leeches, yeah, so anticoagulants. So there were interventions back then. We've gotten much more sophisticated with all of this stuff. Today, there are 18,000 different diagnostic tests that are approved for use on humans. There are 12,000 named diseases, and I don't know how many types of interventions there are. I couldn't really find a source, but there's quite a number of drugs. There's about 1,000 approved by the FDA for drugs, but there's lots of different types of surgical interventions. So if we look at the trial and error medicine of today, the average person sees 4 doctors a year. Each visit is approximately 30 to 50% effective, depending upon the type, and they're supposed to select from what's grown in this gigantic space to be able to diagnose the disease and choose the right tests. And what we end up with quite often is people that wander the healthcare system and don't get a diagnosis for very long periods of time, or they are on treatments that are ineffective, and it can take them a decade, like in rheumatoid arthritis, to find the correct treatment. Okay? And the process we use to get there is largely trial and error. Okay? Well, you know, I don't know if you noticed, but when you use the web now, it's like they know what you're going to buy and they pop it up in front of you, and they're using data-driven shopping, right? They're designing and personalizing websites for you. Why can't we do the same with all of this information we now have to better select treatments and to better diagnose disease? So that's where we're heading. And the other thing that's going on is in the video, the cost of that treatment was a goose, right? And now you pay more than a goose, right, for a surgery. You know, a cancer treatment is upwards of $100,000 today, and we're spending about $3 trillion a year. So there's the promise of producing better results for that money, or even getting people into the right treatments earlier and reducing health costs. So let's move forward. We want to move from a trial and error based medicine to an informatics driven, prediction based model with better outcomes. And so the idea is now, for a relatively cheap cost, if someone were to spit into a tube or take a blood biopsy, we could sequence all 3.1 billion base pairs of your genome and then use that to help decide how you would be treated. Okay, and now I will say that we're at the onset of this, and that it's going to grow and its impact will grow over time, but there is still quite a bit of, in my opinion, current evidence with enough variance where there's probably already value in starting to do this on almost anyone. That said, there are a lot of scientists that are much more conservative than me and think that this is going to take a longer time coming. I think realistically, over the next decade or two, this is really going to move from its current state, where it's becoming the standard of care in cancer therapy and a few other isolated instances, to becoming much more pervasive throughout society and medical care. Okay. So let's go through some examples. So for cancer now, recent research has kind of alluded that there are about 20 different molecular pathways that drive most cancers, regardless of what tissue, and that most tissues may have, several of these pathways, so that you have, just because you have like a breast cancer, there's at least 3 or 4 different types now that are based on different molecular pathways. And so if they're different molecular pathways, they need different drugs and different treatments. So like, it turns out that of the 1,000 drugs that are approved by the FDA, there are over 100 that have on their label that you should genotype and know the sequence of a particular gene in a person before you give that therapy, so you know whether it's going to be effective or not. So in cancer therapy, like if you were an EGFR positive cancer, which is a particular gene, you would treat with Herceptin. And in cancer therapy now, if you're not doing this, you're really not practicing what's the current standard of care. So this idea of personalized medicine is already making its way into mainstream medicine. Moving down for prevention, I know this rings true for at least one person in the room, but you could be very susceptible to cholesterol deposition and heart disease, and so this could be used to decide which statin to give. And I'm just using that as an example, but there are lots of other ways it could be used in preventative manner for your healthcare. For disease diagnostics, there are about 5,000 or 6,000, I think 6,000 single gene disorders that are rare in the population, but collectively about 1 in 10 to 1 in 20 people have one. I have one, it turns out. And you can identify these by sequencing one's genomes, and since they're rare, sometimes they can take a very long time to identify. And in fact, there's formed a national network for undiagnosed disease, and early studies in this program have shown that in 30 to 50% of people that walk in the door and don't know what they have, but they have symptoms, they're able to get a diagnosis for what disease they have, and sometimes get a treatment. So another way that the genetics is starting to make its way into healthcare. pharmacogenetics, I'm going to give a few examples. The fourth leading cause of death in the US is adverse reaction to drugs. So it turns out that a small percentage of the population has a very poor response to some drugs, and it can result in death. I had one actually in the past few years to something I took. I'm still alive. But an example, I'm not going to give an example, but that's one case. Then you have enzymes in your liver that break down drugs. But we all have different alleles in these enzymes. They're called P450s, and people are either fast metabolizers, medium, or slow, and depending upon which one you are, which can be determined from your genetics, that determines what dose of drug you should get. If you take too much, it could, cause adverse reactions again. So that's called pharmacogenetics, and this is important as well in healthcare. It's also finding its way into other things. I've found example of surgical genetics, where people with different genotypes respond differently to surgeries. Nutrigenetics, designer diets. There are some variants that are emerging in this area. It's used in animal breeding, and it's finding its way into society in many different ways. Okay, so over the past 150 years, the life expectancy has doubled, and basically a lot of this is probably, although Ross will argue with me, a lot of this is probably due to biomedical and biological sciences and our practice of medicine. And when you listen to the media, sometimes the role that scientists played is pitched negatively in the media quite often. I even hear it in my own home, that people don't trust scientists. But it's probably the worst marketing effort in history, that we have a group of people that collectively have had such an influence on society and are not really perceived very positively for this contribution. And I think that's a shame. And so the question is, moving forward in personalized medicine, if we were to multiply like the budget of the research fivefold, and could increase the life expectancy by better connecting all these variants to actual real life situations where you could use this in medical care, wouldn't that still be worth it to you? And it's really not a big influence on the annual budget of the US. And that's my final pitch. But what I tried to do today was just give you kind of an overview of what personalized medicine is and how it's beginning to influence our society. And I expect that over the next couple of decades, you'll all be using it in one way or another. So thank you for listening, and I'm happy to answer any questions.
Yes, so right now, what would it cost to do a full sequence on a human?
Okay, so the question is how much does it cost today? So there are two ways to answer the question. There's a cost of how much would it cost our lab, like in the Nevada Institute of Personalized Medicine, to do it, and then how much would it cost to do for you in a more clinical sequencing setting. For our lab, what we do is we're interested in doing the genome that codes for proteins, because that's the real important region, and to do that it's about $700, but to do the whole genome it's about $1,500 today.
3.1 billion.
Yes.
And what's the time frame to do it?
Well, it depends on the lab, but it's less than a month. To actually do it, you're going to run it in, depending on the machine, overnight. Okay?
And that cost is greatly diminishing?
Well, so the clinical is going to be more expensive because there's liability involved. The cost has been decreasing very rapidly. I think, we're going to start hitting a point where, you know, the goal is to do the genome for $1,000, and it's been a goal for a while. And, you know, to run a panel now, like, let's say you had like a cardiomyopathy and they were going to run a genetic panel on you. The cost to run just a panel of maybe 5 genes or 20 genes is, you know, $1,500 to $2,500. So I think they're trying to get the cost into the range of what's already going on. And then instead of maybe getting a panel done in the future, you'll just have your whole genome done. And then you would still have to go back. There's an error rate in it. And then you confirm by a more gold standard method anything you find, just to make sure that you have that. That's called Sanger sequencing. Okay, so we're gonna, before we go on with questions or a discussion, we're gonna take a bio break.